Swayam Prabha. Digital India. Educated India.
Good morning. I am Srividya. I am an associate professor in the Institute of Anatomy, Madras Medical College in Chennai, Tamil Nadu. In this session, today, we are going to discuss about the microscopic anatomy of large intestine and appendix. The objectives of this session is that the first year students at the end of the session will be able to describe the 4 layers of the large intestine, the cells specific to the mucosal layer of large intestine as compared to the rest of the intestine, the differences between the microstructure of the large intestine and the small intestine, and the structure of the muscularis externa. Before we proceed with the session, we need to know why we should know about it, namely the applied aspects. So that is directly correlated to the functions of the large intestine, which we will be seeing in the next slide, and the diseases that can occur, namely the ulcerative colitis and the carcinoma, which we will see in detail in the later part of this session. Any histology or the microanatomy that we tell of any structure is basically designed to ensure that it functions appropriately. The structure's function determines the microanatomical feature. So we will start with the functions of the large intestine. Most important function that we all know is the absorption of water. Whatever is ingested is absorbed, the nutrients, the water, everything is absorbed in the small intestine, mainly in the jejunum and ileum, and whatever undigested material along with the liquid, that is water, that enters the large intestine part is converted into a solid feces by absorption of excess water, thereby ensuring that the body retains the maximum water. So the main function of the large intestine is to absorb water, thereby converting the liquid undigested material to solid feces. It also has a role in the production of vitamin K and B12. Before we go into the microanatomical structure, let us have a brief overview of the general structure of the large intestine. This we know. This is the ileum, which is joining the large intestine, the terminal portion of the ileum. So here is the dilated part of the large intestine, which is the beginning, which is the cecum. And from the cecum, you see a small projection here. This projection is the appendix. Appendix is an addition. So an additional structure projecting from the cecum is the appendix. From the cecum is the ascending colon, the transverse colon and the descending colon, followed by the sigmoid colon and a dilated rectum and straight. Recti means straight. So the straight portion which is dilated is the rectum, and of course the terminal portion of the large intestine, namely the anal canal. So these are the parts of the large intestine. The large intestine grossly, if you see, it is again made up of muscles. It's a muscular bag which is lined by mucosa. That's what is the foregut is made of. The foregut is made up of lining epithelium. I'm talking about the embryological part of the GI system. The gut tube is lined by endoderm and it is covered by the splanchnopleuric mesoderm that gives rise to the muscles. Okay, over the muscles is the serosa, which is in places where peritoneum is present, you call it as serosa. Wherever peritoneum is absent, we call it as adventitia, because adventitia is connective tissue. So if you take the large intestine, the same layers, mucosa covered by muscle and still more covered by serosa in transverse colon. Transverse colon and sigmoid colon are suspended by the mesocolon, whereas ascending and descending colon, they are covered by peritoneum only in their anterior aspects. Posteriorly, they rest on the posterior abdominal wall. They don't have a peritoneal covering. So, if you take a section there in ascending colon or descending colon, we may not see the outermost serosa in the posterior aspect. We will see an adventitia, which is the connective tissue. With regards to the muscular layer, in rest of the small intestine, of course, stomach again there is a small change. If you see small intestine, it has 3 layers, is it not? A circular layer and a longitudinal layer. Okay. In large intestine, the outer longitudinal layer is modified in such a way it is called as tinea coli. This grey structure that is seen is the tinea coli. Then we have the sacculations, that is the predominant, the most striking feature of large intestine. You can see the various sacculations and the yellowish structures called as appendices epiploicae. Appendices epiploicae. Appendix again because they are all extra projections, appendix, addendums, additions. Okay, epiploicae. They are filled with fat, that's why they are yellowish in color. So, the tinea coli, appendices epiploicae and the sacculations are hallmark features of large intestine in gross view, in a gross naked eye examination. In a microanatomical view, that is under a microscope, we see the large intestine like this. The large intestine has a mucosal layer, this is the lumen, the mucosal layer, and then the submucosa, then the muscular layer, and then the tissue, the adventitial tissue. So, this is the mucosa, submucosa, the muscular layer and the serosa or the adventitial tissue. We will see in detail about each one of these layers. The mucosa of the large intestine, this is a hyper view that we are seeing. The mucosa of the large intestine, you can see it is lined by simple columnar epithelium. By simple columnar epithelium, what we mean is it is one layer and the columnar epithelium is simple, it does not have any villi or microvilli or anything. So, it is a simple columnar epithelium. In between the columnar epithelium, in the lamina propria, we see the crypts. We see these crypts, which are the tubular glands that are present in the lamina propria. They form the crypts and they are called as the crypts of the Leberquin. The cells that are present in the simple columnar epithelium, though we call it as columnar epithelium, there are 2 types of cells which are present in the epithelium. One is the columnar cells, the main columnar cells, because the function of the large intestine is absorption of the water, they are called as columnar absorptive cells. And the second type of cells that are present are the goblet cells. In this picture, you see the white pale cells, empty cells, those are the goblet cells without anything inside. That's because the goblet cells in general, when they are in alive persons, it is filled with mucin. Whenever we take the section and then process it, the secretions, the mucous secretions are all emptied away because of the processing, so they appear empty. Now, if we want the goblet cells with the mucous secretion to be visible in microscope, we need to do a special stain which will take up the mucus. Okay, here in a routine H&E staining, that is hematoxylin and eosin staining, the goblet cells appear empty because the mucin has been, which is present, which is filled within the goblet cells, is in process it disappears. So we have the epithelial layer lined by simple columnar cells and the goblet cells. There is an additional third type of cell which is present, that is caveolated tuft cell. Okay, they are seen very rarely. Now what are these cells? We will just see in detail. The columnar cells, because their main function is absorption, in the lateral membrane, in their cell membrane, there is a pump mechanism called sodium-potassium ATPase. The sodium-potassium ATPase is present in the lateral aspect of the plasma membrane. It pumps out, the pump functions in such a way that the water that is present in the lumen is absorbed and circulated into the system. Okay? So, the main function of the columnar cells is to absorb the excess water present in the lumen of the large intestine, and that function is enabled by the presence of sodium-potassium ATPase pump in the lateral aspect of the columnar cells. What is the function of the goblet cells? The goblet cells, as I told you, they secrete mucin, and the mucin helps in the smooth movement of the feces through the small intestine, because as the water is absorbed, the liquid undigested material becomes more and more solid as the material goes deeper into the large intestine parts. So, from the cecum and ascending colon, by the time the undigested material reaches the rectum or the anal canal, it is through and through, it becomes a solid material with all the water that is required getting reabsorbed. So, to make this passage of the movement of the solid material through the mucosa, through the lumen, the goblet cells secrete the mucin, which facilitates the lubrication, thereby allowing free passage of the undigested solid feces. So, what is needed? The mucin has to be secreted more in the lower parts of the large intestine, in the distal parts of large intestine compared to the proximal parts, because proximal parts more of water will be there. Only as the undigested material is moving to the distal part, after the water is reabsorbed only, it becomes more solid. So, the goblet cell ratio also keeps increasing as it moves. So, if the ratio of the columnar absorptive cells to goblet cell is 1:1 in ascending colon. So, for every columnar cell, there will be only one goblet cell, whereas as it moves, it becomes more and more, and the ratio becomes 1:4. Okay? So, the goblet cells are more in ascending colon, and then as it reaches the rectum, the goblet cells become lesser, because there the need for propulsion is less, and already it has been propelled towards the rectum. So, the goblet cells, the ratio of the goblet cells to the columnar cells is more in the proximal parts and less in the rectum. With respect to the caveolated tuft cell, many of the literature say that they are all actually goblet cells only, but then they are caveolated, that is, they are appearing empty, or they are because their secretions have already been expelled, and what we see is the apical portion of the goblet cell without the secretion. So, that is why they appear caveolated. So, literature says that these cells that are present rarely are all actually modified goblet cells without the secretion, or rather exhausted their secretion. So, this is the mucosal layer. Next, let us look into we have seen the simple columnar epithelium in the mucosa, the cells of the simple columnar epithelium. Now, we will also have a few words about the lamina propria, which is on which are the simple columnar epithelial cells that are resting. in the lamina propria, there are numerous lymphocytes and endocrine cells. Now, this endocrine cells, stem cells are all features of the small intestine also. They are all grouped as amine precursor cells. They are present in the GI system. They have their paracrine functions. They function as hormones, not necessarily in this area. They are secreted into the blood and they go to a distant area and they function. So, they have the functions just like any other cells of the same endocrine cells in the small intestine, rest of the small intestine. With regards to the lymphocytes, we can see that in the epithelium, in the lamina propria, lymphatic follicles containing M cells are present. Now, this again, the lymphatic follicles you have seen in the ileum also. They are called as Peyer's patches there. The function of the lymphatic follicles is to ensure that phagocytosis is done of the microorganisms. Now, these lymphatic follicles are many in large intestine because these are the areas which come across the microorganisms as a first event compared to the rest of the system. So, the lymphatic follicles which are present in the large intestine are almost the same as the ileum. They also contain M cells and their function is to phagocytize the microorganisms. In fact, they are called as GALT, gut associated lymphoid tissue. So, in the lamina propria, that is in any part of the mucosa, we see the columnar, we see the epithelial cells, which underneath to the epithelial cells are the lamina propria. So, in the lamina propria, in large intestine, we see just like other parts of the intestine, we see a lot of endocrine cells, stem cells that give rise to more columnar cells as and when necessary, and lymphatic follicles which are called as gut associated lymphoid tissue. Classically, if you compare the rest of the small intestine to the large intestine, the villi are absent. In small intestine, the hallmark feature is presence of villi. In large intestine, it is a simple columnar epithelium. There is no mucosal folds, no villus structures, and Paneth cells are also absent. So, this is what differentiates a large intestine from a small intestine microanatomical feature. Absence of villi, absence of mucosal folds, and absence of Paneth cells. The mucosa is smooth with crypts of Liebekind or tubular glands, and these tubular glands, in the tubular glands, we see these many cells which are like any other cell in the rest of the intestine. Now, one peculiar feature of lamina propria in large intestine is they have outside to the epithelial basement membrane, there is a collagen layer which is present close to the blood vessels. The blood vessels course through the lamina propria. Now, between the blood vessels and the basement membrane of the columnar epithelial cell, there is a collagen layer, and this collagen layer is responsible for the transport of the water. From the lumen, we saw that the water gets reabsorbed and goes into the back to the vessels, and that reabsorption happens through this collagen layer which is present outside the basement membrane between the blood vessel and that is given a specific name called as collagen table, which is outside. Apart from that, we also have some lymphatic vessels generally in lamina propria, whereas in large intestine, the lymphatic vessels are absent in the lamina propria. In rest of the small intestine, the lymphatic vessels are very much high in lamina propria because they have to take the nutrients that are absorbed from the lumen into the portal system. Whereas in large intestine, the lymphatic vessels are absent in lamina propria. They may be present in submucosa. They are present in the rest of the intestine, large intestine, but in the lamina propria, the lymphatics are very minimal in large intestine. Outer to the mucosa, we have the submucosa. The submucosa contains the fat cells, mucophages. Apart from fat cells, mucophages are present. The mucophages are PAS positive granules. The mucophages are phagocytes with mucin. Okay, phages is, it's termed as mucophages because they are phagocytes filled with mucin. So whenever the mucosa, as the feces traverses the pathway, the mucosa may be mildly damaged and that may release the mucin, and that mucin is phagocytized by these cells. So they are mucophages which are present normally in the large intestine. Though they represent a mild mucosal damage, they do not have any clinical significance. Coming to the muscularis externa layer, we already saw that unlike small intestine, here it is organized as 3 thick bands. The 3 thick bands are called as tinea coli. They are the outer longitudinal layer. Instead of presenting over the submucosa as a single layer, they thicken to form 3 thick bands, and in between the thick bands there are thinner membranous structure of the same muscle, the longitudinal muscle, but it is very thin. So what is more apparent is the 3 thicker bands on the histological section and also in a gross view. They are called as tinea coli. Now with respect to this tinea coli, one other interesting thing is the bundles of tinea coli. Within the bundles of tinea coli, the circular layer, we said no, there is an inner circular and then there is an outer longitudinal. So from this outer longitudinal tinea coli, some muscle fibers in regular intervals, they intersperse and get inside the inner circular layer. So thereby they form a mild attachment to the inner circular layer. So wherever these fibers have penetrated the inner circular layer, then these parts become more, the parts in between these insinuations, wherever some parts of the circular layer is getting infiltrated by the outer longitudinal layer. So the portion of the large intestine between the insinuations is acting independently. It will contract independently. In fact, that is what is causing these saculations. So the hallmark feature of hastrations, which we call it as saculations in the large intestine, is probably due to the presence of these longitudinal tinea coli fibers penetrating the inner circular layer at intervals, causing independent segments of contraction, which are the saculations. Okay. When we talk about these contraction, because this muscularis externa is responsible for the contraction and propulsion of the feces down into the rest of the large intestine. These contraction are of 2 major types. One is local segmentation, that is, they contract only in that local area. It is segmented. They do not propel the feces down. It is only to move within that segment. The second movement in the muscle that happens is the peristalsis, which happens through the gastrointestinal system. That peristalsis pushes the feces throughout the passage of the large intestine. So, with respect to muscularis externa, the 2 specific features in large intestine is the outer longitudinal layer. Instead of being a single layer, it is organized as 3 thick bands. Between the bands, there is a thin layer. And from this longitudinal layer, some fibers enter deep into the deeper circular layer, thereby causing saculations and causing localized segmental contractions apart from the regular peristalsis. The outermost layer is the serosa, and as already told, over the ascending and descending colon, the serosa are absent. They are retroperitoneal, whereas in the rest of the large intestine, it forms mesocolon, and apart from the serosa, there are pouch-like processes which are filled with fat called as appendices epiploicae. And these appendices epiploicae, they are not present in rectum, anal canal and vermiform appendix. So, except for appendix, rectum and anal canal, all other layers, all other parts of the large intestine exhibit this appendices epiploicae. The histological features of the large intestine, we will have a few words about the applied histology. Now, we also already saw that in tubular glands, there are a lot of endocrine cells of the gut, which are argentafin cells and enterochromophil cells. They are all amines and polypeptides and they act as hormones, not necessarily in large intestine. They are secreted into the blood vessels and they act as hormones in an area away from it. So, they are called as paracrine hormones. Ulcerative colitis is one of the disease. By the name itself, we know it is an inflammation. Itis is inflammation. Colon means it represents the large intestine. So, the inflammation of the colon gives rise to an ulcerative picture, and this is one of the condition which gives rise to a disease of the large intestine causing diarrhea and other features. So, this is a diagnosis which is done based on a biopsy report seen under the microscope. The hallmark feature that is seen is the lymphatic vessels that are usually absent in lamina propria in a normal large intestine, whereas in ulcerative colitis, the lymphatic vessels increase in number. So, that feature is one of the important feature determining the presence of ulcerative colitis in a biopsy slide. In inflammation of the appendix, the vermiform appendix again is one of the important clinical conditions seen in large intestine. It is called as appendicitis. We will see in the next slide about the histological feature of appendix. So, any inflammation of appendix, if it is acute, it has to be immediately removed. Surgically, the appendix has to be removed. To summarize, the 4 layers which are present in large intestine, namely the mucosal layer, the submucosa, the muscularis externa and the serosal layer are just like any other part of the intestine with some changes. The primary one being that the villi, which is the hallmark feature of the small intestine, is absent in large intestine. And the Paneth cells are also absent. With regards to the absorptive cells of the columnar epithelium, there is a very specific modification, namely abundance of sodium potassium ATPase in its structure, which aids the absorption of water, which is the primary function of the large intestine. Also present are numerous goblet cells as compared to the rest of the intestine, which secrete mucin. The mucin that is secreted helps in the easy passage of the undigested waste material. The layer of muscularis externa is also modified as compared to the rest of the small intestine. The outer longitudinal layer is modified into 3 longitudinal thickened bands, in between of which is thinner section. This is called tinea coli, and this is very specific to the large intestine. So with that, we come to the end of the session of large intestine. At the end of this session of vermiform appendix, the first year students will be able to describe the 4 layers of the appendix, the specific feature of the submucosal layer of the appendix, and the structure of muscularis externa. Though vermiform appendix is a vestigial organ, its clinical relevance lies in the fact that it is one of the common organs that gets infected. The clinical condition is called appendicitis, which we will see in detail in the later slide. In gross anatomy feature, the appendix is the extra additional appendage which is extending from the cecum, and it is the narrowest part of the gut. Just like the rest of the gastrointestinal system, it also comprises of 4 layers. The 4 layers are the mucous membrane, submucosa, muscularis externa and the serosal layer. So this is a low power view of the vermiform appendix. The lumen is narrow because this is the narrowest part of the gut. We see that the whole appendix is visualized in a single field, a part of the appendix with the complete lumen. In rest of the intestine system, you don't see the lumen like this because they are all dilated. The section cannot take care of both the sides of the wall. In this section, in this field, we see the lumen with the wall surrounded by the wall. So this is the mucosal layer and this is the submucosal layer. You see lot of lymphoid follicles in the submucosal layer. The mucosal layer is lined by the simple columnar epithelium, just like rest of the large intestine. It is simple columnar epithelium, but then it does not have those features unlike large intestine because the absorption of water does not happen here. There is no functional significance for the appendix. So the modifications are not as much present in the rest of the large intestine as in appendix. But just like in large intestine, numerous globular cells are present in the simple columnar epithelium. In submucosa, many number of lymphoid follicles are present, which are the hallmark feature of the vermiform appendix. The abundant lymphoid follicles present in the submucosa is the hallmark feature, which is much more than the rest of the large intestine. With respect to the muscularis externa, it will be a uniform longitudinal and circular layers that are present, unlike large intestine, where the muscularis externa is modified into 3 bands of Taenia coli. The 3 thick bands of Taenia coli are absent in appendix. It is also absent in rectum and anal canal. But here in the section, you can see the uniform muscular layer of circular smooth muscle and longitudinal smooth muscle. Outer to the smooth muscle is the serosal layer. Again, it is suspended by a mesoappendix. So, it's called as serosa. It is a peritoneal covering.
As told earlier, appendicitis is one of the commonest infections that can occur. The narrowness of the vermiform appendix, its blind end, the abundance of lymphoid tissue, the excessive mucin causing impaction, and also impaction due to hard feces are some of the predisposing factors that promote the occurrence of infection. The clinical condition is called appendicitis. Sometimes the inflammation and the infection can transmigrate to the surrounding peritoneum, causing peritonitis, and rarely it can cause perforation of the appendix, both of them being an acute state emergency condition. Now, apart from appendicitis, one other condition that is associated with appendix is presence of carcinoid tumors. Among the sites of the carcinoid tumors, appendix is one of the most commonest.
To summarize, in vermiform appendix, the 4 layers are present, like any other part of the digestive system, namely the mucosa, the submucosa, the muscularis externa, and the serosa. As regards to the mucosa, the number of goblet cells are numerous when compared to the small intestine. With regards to the submucosa, the specific feature of vermiform appendix is presence of numerous lymphoid follicles, which is the hallmark feature of identification of the vermiform appendix. And as compared to the rest of the large intestine, the outer longitudinal layer of the muscle, namely the muscularis external layer, the Taenia coli, which is the 3 thickened longitudinal bands present in the rest of the large intestine, is absent in vermiform appendix. Not only in appendix, it is also absent in rectum. So these are the specific features of the vermiform appendix. Thank you.